So good afternoon. I'd like to thank, obviously, the organizers of OIS for having us here today. I'm going to tell you a little bit more about who we are as a company. We are a publicly listed NASDAQ-traded company, albeit certainly microcap territory. So with that, obviously, I have my forward-looking disclosures and my statements here being applicable to those. So who are we as a company? We are truly dedicated within the segment of orphan retinal diseases. We know that there's still a lot of unmet need within a lot of these different, whether inherited or age-related or inflammatory-driven diseases, and that is where we are truly spending our time and playing. If you take a look at our pipeline, we do have multiple assets, all of which are small molecules. And I'm going to spend most of my time today talking about our Kiora 301 program, which we just read out last month at the American Academy of Ophthalmology's Retina Subspecialty Day, our phase 1b results in retinitis pigmentosa. If you do look, though, as a company, we have a lot of upcoming milestones, including kicking off 3 phase 2 clinical trials, which will be run in the 2024 calendar year, and hopefully in about a year's time, I'll be up here talking about our phase 2 results from some of those studies. So again, jumping into the molecule itself, and again, this is our lead molecule, our lead program within the organization. We are targeting some inherited retinal diseases. As you know, and I don't need to explain it in too much detail to this group, in a lot of these inherited retinal diseases like retinitis pigmentosa, it leads to death and degeneration of the photoreceptors, typically the rods first, peripherally, then moving more towards central vision loss in the cones as well. We know that when that occurs, vision is lost, pretty simple. So what are we doing? Kiora 301 is a novel small molecule based on an azobenzene core that has the capability, in the presence or absence of light, of changing its isomeric configuration. And so it can go from trans, a linear state in its lower energy form, up into a higher energy cis isomeric configuration. We know the molecule gains entry specifically to retinal ganglion cells downstream of degenerated photoreceptors due to the upregulation of a channel called P2X7. And once inside the cell, it resides in a non-covalent way on potassium and HCN voltage-gated ion channels. And again, when light hits that molecule, it changes shape. It alters the signaling directly to the brain, either turning it on or off, and thereby what we expected and were hoping for was actually conferring vision back to patients with later stage disease. You guys know a lot about RP. There's a lot of different gene mutations that can cause this disease. There are gene therapies in development today to try to target individual therapies or individual mutations. What we like about our approach is that it is truly gene agnostic. So jumping into, there we go, a little bit of just background data. There's a lot more in our non-clinical and research packages, but we can show that with a retina from an animal that has a transgenic mutation causing retinal degeneration, you can directly correlate light switching on and off to electrical activity coming out of that retina. And then when you apply it into different behavioral studies in animals, you can actually give them back the ability to perceive light and have a behavioral change associated with that light being on or off. With the rest of the data, a lot of safety data, obviously we kicked off this phase 1b clinical trial. We are starting with patients that are off chart, so really going for the worst of the worst patients and assessing a host of obviously safety tolerability endpoints as well as efficacy endpoints. I'm going to show one patient testimonial. I promise there won't be too many of these, but we do have these from every one of our patients in the study. This is one that's just really compelling to me. Is there a way to start the video? Maybe.
Later that week, I went back home to Melbourne, and one of my hobbies is bike riding. And we go out bike riding early in the morning, often leave when it's first dawn or dark. And as part of that, I put the lights on the bike, and and on the tandem bike, I've always relied on someone else to say, is the light on? Is it flashing? And because I had no concept myself, and I sort of looked at this light and turned it on and go, hold up, I can see that light, and I can see whether it's on or whether it's flashing. And I think that, you know, that was the first time, not only in a clinical sense, but actually it really goes, wow, this is really useful.
This was an individual who for about a decade has been NLP, truly unable to see any form of light, recognize anything. So having him come back a few days after this injection and telling us these types of responses, we felt was incredibly exciting and encouraging. So again, small study, we have to innovate somewhere, and you can't do it with these massive studies, but this was the right one to do at the time. What we also looked at was kinetic visual field, so using essentially a modified Goldmann, and we surprisingly saw statistical significant increases in the visual field itself in those cohort 2 patients. So those are the ones that have hand motion or could have count finger visual acuity. We looked then at visual acuity, specifically on both cohorts of patients, the very severe and then just the slightly less severe. And now what we saw were some actually trended increases in light responsiveness, being able to perceive that light over the 1-month period of time that we assessed. And in the better seeing patients, we actually saw about a 0.3 logMAR improvement using the BRVT, a validated visual acuity assessment tool for these off-chart patients. We put them through a host of functional tests as well, and while there's a lot of learnings to take from this, we did see some, again, trended improvements. And what really gets me excited is seeing you have this concordance of response. Multiple different endpoints are showing up increases in activity that then starts to tail off, consistent with the pharmacokinetic profile of this small molecule. We built in functional MRI, and you can see specifically within that striate region of the visual cortex, there is an increase in activity at day 2, incredibly quickly, as well as even at day 14. That tends to tail back off around day 30. This was a big question to me because if the brain typically uses Java, for instance, to interpret the code from the eye, we knew this approach of vision restoration would be basically giving it C++. So can the brain actually use that in a functional way? So the key takeaways are, I didn't show you the safety profile, but I can tell you it was incredibly uneventful. There was no retinal inflammation at all observed. It appears to be proving the mechanism of action, and we are going to be taking this now, again, into a larger controlled phase 2 study next year. So with that, I'd like to thank you.